perceived as great. So why not use this editing in an even more fruitful way? This is what CRISPR-Cas9 does. We are simply editing a gene, a person's mutated gene to a normal one so that we can help them grow better. So let's move on to what is sickle cell anemia. It is basically an inherited blood disorder which is caused due to a mutation in HBB gene found in chromosome number 11. Can you imagine 3 lakh infants die annually due to sickle cell anemia worldwide? 20% children with sickle cell disorder in India die by the age of 2 years. 1,000 children in Africa are born with sickle cell every day and more than half die by the age of 5 years. And 1 lakh Americans have sickle cell anemia. And the sad part is there is no permanent cure treatment yet, but some include medications, blood transfusions and stem cell transplantation, where even clinical trials are going on for stem cell transplantation for sickle cell anemia. Disadvantages include risk of complications, limited availability of donors for stem cell transplant or blood transfusions, adverse effects of drug therapy, absence of potentially curative treatment regimen. So basically our aim is to study the advancement in genome editing and overview the CRISPR-Cas9 technology. CRISPR-Cas9, it stands for Cluster Regularly Interspaced Short Palindromic Repeats, which is a revolutionary gene editing tool which enables precise and targeted modifications to the genome. So let's have a look onto the components of CRISPR-Cas9. It includes basically 3 components. One of them is Cas9, which acts as a molecular scissors. Second is a guide RNA, which further includes spacer DNA, which together forms the targeting mechanism. And Cas9 and guide RNA together forms the CRISPR-Cas9, which is the precise molecular surgeon. So how it works? The Cas9 protein forms a complex with the guide RNA, and then this complex attaches to a matching genomic DNA sequence adjacent to a spacer. Then this complex, it cuts the double strands of the DNA, and then the programmed DNA may be inserted at the cut. The specific treatment includes CRISPR-based therapeutic solutions are not in the form of pill or a drug, but then instead some cells of the patients are extracted and the genes are edited in the lab and corrected genes are then re-injected. Permanent cure, CRISPR opens up the possibility of finding permanent cure for a vast number of disorders like sickle cell anemia, beta thalassemia, color blindness, muscular dystrophy and several types of cancer, liver and heart diseases. Results include this approach has the potential to provide one-time cure for sickle cell anemia. Success rate, there's 50 to 70% of repair success rate was found after the treatment, and only 21% of population showed no sign of disease for 6 months after a single treatment. Major challenges include autoimmune response, off-targeting. Thank you. And lastly, I'm very glad to say this that Professor Jennifer Doudna and Emmanuel Charpentier are the founders who received a Nobel Prize in Chemistry 2020.